So if you've tuned into the other sessions, you probably got familiar with me already. I've been the host for the webinar series. My name is Jeff, by the way. I'm one of the strategic account managers for Synthego in the Boston area. So I've got about 7 years of biotech experience, first at the bench and now on the business side of the industry. And my main role at Synthego is to be our point of contact for some of our clients in the Boston, Cambridge area. And by the way, if you ever have any questions about Synthego, feel free to reach out to me if you're in Boston and Cambridge, or if you're elsewhere. I'm sure there's an account manager in your area. We've got a tech support team, or as it relates to today's topic, then a separate clinical or GMP team as well. On that note, let me introduce Abby. So Abby has over 2 decades of experience in the biotech industry at companies like GSK and Thermo Fisher, and currently she is a clinical sales consultant at Synthego, where she supports our clients with their needs for GMP-guide RNA for CRISPR-based therapies and diagnostics. So with that, let me give a quick introduction to today's talk and then pass it over to Abby for the presentation itself. So according to the FDA, there will be about 10 to 20 cell therapies approved per year by 2025. Obviously, CRISPR is a key component of this revolution in treatments and even cures, so the industry is seeing a big increase in demand for clinical grade guide RNA. On that note, Synthego has established itself as a front runner in this area with GMP capabilities. We've got plenty of client success stories and continuous innovations, which I'm sure Abby will talk about in more detail today. So specifically, the topic of the webinar today, or what you can learn today, is we're going to talk about how Synthego can support you throughout your research, whether you're early phase or process development or already in clinical trials with CRISPR-based therapies and diagnostics. And if you are interested in engaging with Synthego on talks regarding GMP-guide RNA, then we can also talk about some clear next steps for how to get in touch with us and have those detailed conversations about your projects. So on that note, I see you've got about 94 attendees, 5 minutes past the hour. So I will pass it off to Abby for the presentation itself now.
Thank you, Jeff. Thanks for the kind introduction. And good morning, afternoon, evening to all the people who are joining us today. We appreciate you taking time to join us for the webinar today around CRISPR-based therapies and diagnostics, and more specifically, how to move along that journey from discovery to clinic with GMP-grade guide RNA. So what we're going to cover today is kind of the journey generally from discovery to clinic, a little bit about GMP CRISPR solutions that Synthego offers, and then we're going to look a little bit at the cell and gene therapy challenges that the industry as a whole is changing, and how we're trying to address those as we move forward. The gene therapy market is probably around like the $15 or $16 billion mark. Many of these therapies are around cell therapy, and more specifically, the cell-based immuno-oncology. There's a fairly nice representation around the gene therapy market, and then just a handful in terms of the tissue and engineering market. And I'm not a big one for normally kind of reading quotes during talks, but this one has a lot of impact, so I am going to go ahead and read it. This is from Scott Gottlieb, who's the former commissioner of the FDA, and he said, by 2025, we predict that the FDA will approve 10 to 20 cell and gene therapy products in a year based on an assessment of the current pipeline and the clinical success rates of these products. So that's pretty astonishing, because what that translates to is that approximately 80% of all of the approved therapies are going to be in the cell and gene therapy space. So that's really changing the way the clinical market is going to look, and how we're treating people who have very serious diseases. So when we talk about cell and gene therapy, not all of that is going to be CRISPR-based. So next, let's have a look at some of the CRISPR-based therapies and where we are in terms of that. So the first CRISPR trial was carried out in 2016. This was using PD-1 knocked out T cells and was targeting non-small cell lung cancer. These trials were done in China. They were followed pretty rapidly by trials that were using hematopoietic stem cells in order to target HIV and leukemia. And then more recently, what we see are companies like CRISPR Therapeutics and Vertex, who are entering the clinic and seeing very promising results in terms of safety and efficacy, targeting the hemoglobinopathies. So in particular, CTX001 is targeting sickle cell disease and beta thalassemia. And then we have other companies like Editas who are starting to enter clinical trials, not using cell therapy or using ex vivo cell therapies like the T cell targeting therapies are, but actually doing in vivo. And the first of these, not surprisingly, are being done to treat eye diseases. One of the reasons for that is that the eye is very easily accessible. So it's easy to access and deliver directly to the eye. This slide says that there's currently around 51 ongoing registered CRISPR trials. This has already grown since this slide was made. And if you just look at a couple of the, I think there's 5 here, of the therapies, a lot of these are targeting the hemoglobinopathy, so sickle cell disease, beta thalassemia, but we're starting to see more and more entrants that are targeting other diseases. So this is kind of the CRISPR-based therapies. Another player that maybe we don't hear as much about, but has become really important over the last year, is how CRISPR is playing a role in diagnostics. So there were several companies, notably Sherlock and Mammoth Biosciences. They were targeting things like trying to detect specific SNPs that are biomarkers for cancer, looking for specific DNA and RNA sequences that would indicate the presence of bacterial or viral infections. And to do this, they're using some of the non-Cas9 based CRISPR reagents, so using things like Cas12a enzyme, which targets double-stranded DNA the same way that Cas9 does, as well as Cas13, which will directly target RNA, as well as DNA. And this became particularly important over the past year because, as we all know, we've been faced with pandemic of SARS-CoV-2, which is an RNA virus. What this meant for these diagnostic companies is that there was a huge influx of money, so they would become very well funded. So a lot of these technologies accelerated very rapidly. 2 of them I'd like to call out are the Stop COVID, which is based on the Sherlock testing system, as well as the SARS-CoV-2 detector kit. These are all being used under the FDA emergency authorization, use authorization, along with the vaccinations that we've all been getting. Basically, the way these work is that you deliver your CRISPR reagents into a tube that has a patient sample in it, along with a fluorescent reporter that has a quencher attached to it. Once the CRISPR reagents find and bind their target sequence, it causes a cascade that will kind of, what happens is the enzyme goes a little bananas and starts cutting up molecules randomly. These reporter molecules get cut, decoupling the quencher from the fluorophore, and you get a fluorescent signal. These are very rapid, so you can get a result very quickly, and in some cases without having to have really complicated equipment. So I spoke a little bit about kind of generally the CRISPR therapeutics and CRISPR and diagnostics. So I want to step back a moment and kind of look at Synthego and who we are and where we fit in this environment. So we are located, if we start up at the top left, we're located on the west coast of the United States in Menlo, California. This is a representation of Synthego. We've grown very rapidly, so we probably would not all fit into that picture anymore. But it's important to understand that Synthego is a CRISPR company. This is all that we do. Everything we do is based around CRISPR. We've done hundreds of thousands of gene edits using CRISPR, not just using Cas9, but also using some of the other CRISPR systems. We have, when we do engineered cells, we get better than 80% average editing efficiency, which is really phenomenal. We have hundreds of publications that have cited us, as well as a fair few publications that we've participated in. We're being used around the world. And we have partnerships with many companies, so in commercial environment, as well as in the university environment and with governmental agencies. So what's unique about Synthego? So we're really trying to lead the way using CRISPR by industrializing it. So what does that actually mean? So if we start over here on the left, we're going to move kind of across and down at the same time. We start with kind of a collection of core capabilities that involve chemistry, biology, automation is a key to what we do. We have proprietary bioinformatics that really has driven our ability to create guide RNAs that have very high targeting efficiency with minimal off-targeting efficiency. We use machine learning in order to gather information and make better decisions about what we're going to do next in ways that really require that kind of computational drive. And what having this kind of core stack of competencies enabled us to do is to build two platforms. Our CRISPR guide RNA-based platform is called HALO, and our engineered cells platform is called Eclipse. What this is doing is it's allowing us to use our automation in order to make large numbers of guide RNAs in parallel at very high quality and very rapidly. And just as HALO allows us to do that in terms of synthesizing guide RNAs, Eclipse is allowing us to do that same sort of industrialized approach in engineering cells. So being able to do large number of cells at the same time, so without having to do them sequentially, with very high quality and very rapidly, we're being able to drive down the cost and really enable people to interrogate more questions at one time. And if it's starting to look like we're building a new stack in this middle here with platforms, we kind of are, because Eclipse couldn't really exist without HALO. So if we didn't have this methodology for making large numbers of guides very rapidly, we wouldn't be able to feed the platform that enables us to make large numbers of cells, engineered cells, very rapidly. And at some point, both of these are going to combine in order to make a third platform that we'll talk a little bit about later on in the talk. From early research and development, so from screening, target validation and identification, into kind of the preclinical and clinical environment, where does Synthego fit into that? And how does that kind of fit into the larger cell and gene therapy environment? So, people who attended the first webinar learned a bit about our modified synthetic guide RNAs and our gene knockout kits and our screening libraries. These are really all designed to help you figure out what is going to be your best target. So which genes are involved in the disease or the phenotype that you're interested in, validating what comes out of those screens, and then identifying which are going to be the most appropriate targets to move forward with. So identifying those leads. Our engineered cells, so in terms of pools and clones, can also be very useful. So once you have some of those candidates, these can really help you do more biological tests to ensure that your target is really worth pursuing and that the phenotypes you're seeing really are due to what you think they are. And then as you move kind of closer towards being able to bring your CRISPR, your idea for a CRISPR-based therapy into the clinic, we have GMP-like guides. So these are research-grade guides that have a little bit more have better purity and a little bit more documentation, that are really designed to be. Oh, my pointer stopped working. Sorry about that. That are really designed to be kind of your IND enabling quality for guides, and then moving into GMP guides. I'm going to talk about the difference of these a little bit in a little bit greater detail as we move forward. But what we're finding in talking to regulatory agencies is that they really are requesting GMP guides a little bit earlier than most people expected. They are being considered a drug substance. So even in the cases of cell therapies where cells are going to be the drug product, it's starting to look more and more like people are going to have to switch to GMP a little bit earlier than they'd hoped. So these two grayed out areas, so these are process development cell lines and GMP cell lines. And these are going to be the things that are going to start making up that third platform stack I alluded to earlier on. So we are hoping and planning to be able to help facilitate moving into the clinic by providing cells.
Abby, before you move on, do you mind if I jump in with a comment on this slide?
Not at all.
Cool. So yeah, I mean, I think this slide really sums up what we've been trying to highlight with this webinar series, where no matter where you're at on the discovery to clinic spectrum, there's probably something that Synthego can offer as support. And really researchers engage with us at any stage in this process. So certainly some people do take the logical path of first engaging with us through libraries and doing screening, and then doing more thorough disease modeling with engineered cells, and then finally on to clinical applications with GMP guide RNA. But some people work backwards too, where maybe they first work with us on GMP, but then they've got some other programs, and then they come back to us for those more research products. So really, I mean, if you're doing any type of CRISPR work, particularly screening or disease modeling or these CRISPR-based therapeutics, definitely reach out. If you're kind of in that GMP phase, you'll probably get in touch with Abby or someone else from our clinical team. Or if you're more on the early side of discovery with screening libraries or disease modeling, then you'll probably get in touch with me or one of our other account managers for more research-based products. But in any case, really trying to support that whole workflow, no matter where you're at. So yeah, look forward to hearing more about your projects after the talk here.
Mm-hmm. Thank you, Jeff. So we are going to move towards more towards the GMP CRISPR solutions. We're sort of going to revisit this slide in a little bit more detail later on in a different context. So I'm happy to move on from here now. So now my mouse, there we go. So in terms of GMP CRISPR solutions, we'll start with our facility. So if we start at the top, so we do have a state-of-the-art GMP facility. This is a picture of the actual facility. We've got blurred people because they were working when this picture was taken. I'm sure it was done through glass because we're not allowed in that facility. It is ISO 9001:2015 certified, so we received that certification a little bit later than we would have without COVID because everything was delayed because of that. We're in full compliance with the international standards, so our rooms, we have these regions, that are ISO 8, ISO 7. We've got an ISO 5 biosafety cabinet in here. So full compliance going on with our facility here. We started making GMP-like guides near the end of 2019 and launched full GMP at the beginning of 2020. We were able to deliver our first GMP guide within the first half of 2020. And despite only doing this for about a little more than a year and a half, we've completed and delivered more than 60 GMP-like and GMP campaigns so far, and continue to see keen interest from people all over the world who want to be able to bring their CRISPR therapies and diagnostics into the clinic. So we're trying to support CRISPR programs, and we're trying to help you get to the market faster. We also recognize, though, that I'm going to start over here kind of on the right side of the slide to start with. We recognize that, as you're in your research phases, so you're in your preclinical discovery phases, that you're going to need probably lots of guides, but at very small scale. You're not going to need GMP. As you move into early IND enabling or early phase 1, you're going to be engaging, a couple of patients, a handful of patients. Cell and gene therapies in particular in early phases tend to enroll very few patients at a time. These are often targeting rare diseases, so there aren't very many people available to enter these trials. Or, we're also being very cautious because these are very new. But as you can see, as you progress through this kind of journey from bench to clinic, you need fewer different guides, but you're going to need larger amounts of each guide as you kind of traverse this terrain. And we recognize that, and we're planning our scale up so that we'll be able to kind of march right along with you as you progress. So as of now, we're perfectly able to take you from early research into GMP. We can help with analytical development. We have dedicated project management. We have a full quality control and quality assurance team. We're able to do about, I would say, 10-ish grams a year right now. We're planning to be able to expand that somewhat by the end of this year, but we're planning a significant, so 12x expansion in next year, so that, we're taking this 10 grams to 120 grams per year. And we're already talking about the next expansion after that, because as I showed you in that early slide, more and more of these programs are going to be entering the clinical space, and we want to be able to support folks both as they enter, so be able to do large numbers of different guides at the GMP grade purity, as well as large amounts of individual guides as necessary as you progress through that. So not only are we focusing on guide RNAs and engineered cells, so if we start again over on the right here, you can see, this, I call this the peace sign pie chart. Our guide RNAs support our HALO, our engineered cells are our Eclipse platforms, but we're also working on new technologies that are going to make kind of that third platform. We're definitely listening to what people are telling us, and we're developing things that should help meet some of the upcoming needs. Some of these include CRISPR on and off. So this is a temporal control for controlling when CRISPR works. So you can deliver it, you can turn it on and then turn it off very rapidly. This is very useful in terms of controlling potential for off-target effects. We are exploring novel nucleases. We're able to make guide RNAs for multi-nucleases, both at the research grade and the GMP grade levels. We're looking for optimizing and potentially making GMP ribonucleoproteins, so to take that complexation step and bring it into kind of a more regulated GMP environment, so that rather than delivering GMP grade guides to you and having you complex it, we can deliver that to you already ready to use and fully GMP compliant. And really starting to explore the idea of developing a high throughput and cost-effective GMP cell manufacturing facility. So in the hopes that at some point we'll be able to actually support you when you need to have somebody make your cell therapy for you. So how can Synthego support you in terms of your guide needs as we move from research, which is really ideal for kind of this early discovery phase, where you're interrogating, so maybe you found your gene target, but you want to try to find the specific guide that works best against your specific gene, so that you can bring that forward as part of your therapy. And we would recommend that you use research grade guides for that. As you start to narrow down those numbers and you're down to like 2 or 3, you might want to move into something like a research grade plus a little bit better purity. So something like an RUO plus HPLC. I probably should have said previously, RUO is research use only. What I really mean when I say that is research grade. And then as you're starting to do kind of like your pre-IND or your IND enabling studies, probably want to move into using a GMP-like guide that has a higher quality profile, so a little bit better purity. It's starting to add some of the documentation that regulators want to see. However, not quite as extensive as what you would expect from a full GMP grade guide, which is going to be fully compliant and have all the documentation that's required in order to move into the clinic. So I'm going to take a minute and talk a little bit more about the differences between the different grades of guides, and then we'll talk about when you might want to choose which guide for which applications. So I would say and if you start on the left, you see the different kind of application areas. So what you're using these for and what you're going to get, and then comparing GMP-like and GMP. So in terms of application areas, we would expect this to be used in preclinical. We're seeing a lot of people using GMP-like guides under the emergency use for diagnostics. So COVID really made a lot of people interested in getting GMP-like guides as quickly as possible for diagnostic kits. We would expect that as therapeutic and diagnostics both kind of move into later phase. So once you're submitting for your IND or you've got your IND approval, once you're going to humans is kind of the real cutoff for this. Really have to have GMP grade guides. The people I've been speaking to who are using these in diagnostic kits do expect to have to make that conversion once the EUA has been stopped. So once it's no longer in effect, we would expect that when you're doing CRISPR-based therapeutics, the GMP-like material would be useful for toxicology studies, for animal studies. Again, as soon as you're moving into humans, really need to start moving into GMP grade guides. The regulators really are, I'm sorry, I did not mean to do that, really are indicating that people are going to have to switch to GMP even for cell-based therapies. The GMP-like guides are made, and again, I'd like to reiterate that GMP-like is still a research grade guide. The reason for that is it's done in a controlled but an unclassified manufacturing environment. So these are not made in a clean room, whereas GMP is made in a fully compliant clean room. It's meeting all of the international standards. There are SOPs in place for changeovers, which we'll talk about a little bit more, and those sorts of things. The raw materials going into these are different. So with GMP-like, they're traceable and customizable, whereas with GMP, they're traceable, and we're doing identity testing to ensure that they really are meeting the standards that are necessary for input into a GMP product. Their synthesis and process controls are a little bit different. So both molecules are being made once we move into larger scale on ACTAs. However, the GMP equipment is, of course, fully qualified and validated. And then a significant difference between GMP-like and GMP is around documentation. So the batch record, so GMP-like does have a batch record. However, it's not as extensive as GMP. So the GMP really is designed to be compliant for your module 3 submission. And we do ask that clients approve those prior to our starting synthesis. So if we move into some of the controls in terms of GMP, We have full cleaning validation, environmental monitoring, and changeover. What that means is that we're ensuring that there are no drug substance product contaminants carried over from one synthesis to the next in any of the equipment, despite the fact that we have no bacterial, fungal, or animal based inputs going into our molecules. So these are fully synthetic. We do test for endotoxin and bioburden. We double check and make sure there's no carryover residue from cleaning reagents, and all of these are validated methodologies. Environmental monitoring of our clean room includes continuous temperature and humidity monitoring. I lost my pointer again, sorry. And then changeover follows stringent SOPs, including line clearance, room release, and also the equipment cleaning. So process controls include environmental controls and GMP and production controls. I'm not going to go through each of these, but you can see very clearly that GMP does have a much more stringent requirement for environmental and production controls. And again, you know, GMP is done in the clean room, whereas GMP-like is not. In terms of production controls, all of the reagents being put into GMP are QC tested. Anything that is not already GMP compliant, we are testing ourselves in order to ensure that they can go into a GMP product. There's full quality assurance documentation review. We're retaining samples, and I think I talked about change control and validation already. There's also full sample chain of custody. So what this means is that every time a reagent is being used, it's being fully documented as part of the batch record. So how does the documentation differ? I told you that, you know, GMP-like and GMP quality is going to be very similar. A large part of what's different is going to be in terms of how we document these. GMP-like does have a batch record. It's a bit more generic. It's actually considerably more generic than the GMP batch record. It includes a certificate of analysis, a final report on quality assurance and quality control. There often are no product specifications that are agreed upon upfront for GMP-like. Often we've never worked with these molecules previously, so we don't really know what those product specifications should be. We don't require pre-approval of the batch records for GMP-like. And of course, any quality events, if applicable, will be reported for both. And the GMP-like really is designed to be quicker and lighter, as well as more cost-effective. For GMP, the batch records are going to be much more extensive. They'll include the certificate of analysis and the certificate of conformance. We've got quality assurance and quality QC oversight throughout the whole process, and this is fully documented in the batch records. There are product specifications and a quality agreement that has to be agreed upon up front. We require pre-approval of the batch records. And these are really designed to take you into your IND submission and be fully CMC module 3 compliant. And I think I've talked about these already. So quality events, chain of custody, product specifications are all going to be documented there. So if next we have a look at kind of where each of these fits in that journey, sort of from early research through clinical trials. This is kind of a changing environment because regulations still aren't fully understood. We've sort of, if you look over here on the left, we've sort of separated this into ex vivo applications. So these are applications in which cells are edited and then the cells become the drug product. So that's the actual therapy, not the guide RNA itself. Or the CRISPR reagents themselves, and in vivo applications in which the CRISPR reagent is the therapy, and also what kind of your risk tolerance is for each of these. And again, it's like when you're pre-IND, you may be able to get away with large-scale RUO. For ex vivo applications, for in vivo, probably not, unless you're really early phase. We would recommend for most of this that you use a GMP-like guide pretty much up until you're ready to enter and submit for your IND. If you're using in vivo applications and you really are very risk averse, some companies are choosing to use GMP throughout this entire process. You can get away with GMP-like possibly for some of your tox and safety studies, your animal studies, things like that. But as soon as you're entering phase 1, as soon as you're talking about putting these into people, you really need to be considering a GMP molecule. We do expect that the FDA is going to request cap closure integrity testing, sterility, stability testing. I'm going to cough, hold on. Sequence verification. And they've asked for a pretty good understanding of what the non-full-length product impurities are in those molecules. So I talked a little bit about what's some CMC controls in here. So let's have a look at what we offer. Excuse me. So on the left here is our standard release testing offered both for GMP-like and GMP-grade guides. They're going to include appearance, purity, concentration, mass, determination. pH, endotoxin, and bioburden are optional. We're starting to hear from, I've got a tickle in my throat. So it sounds like I'm going to start crying, but that's not really the case. It's just this tickle in my throat. Sorry about that. What we're starting to hear from regulatory agencies is they really would prefer to see sterility testing even for GMP-like molecules. That could also mean that we have to do some additional filtration steps. But for GMP, we can do stability studies. We do method development and validation of bioburden and sterility or other tests as requested. We can look at elemental impurities, residual solvents, and all of these other tests here. It is important to note that we can also assist with technical writing and IND submission support. So we have attended some pre-IND meetings with clients so that we can be available to answer any questions. So fortunately, I'm almost done. So the fact that we don't have a whole bunch of time isn't all that urgent. I just kind of want to go very briefly through some of the industry challenges and how Synthego is hoping to help mitigate some of those. If I can make my slides advance. So this is out of Cell and Gene earlier this year. The top 6 most cited responses regarding cell and gene therapy manufacturing needs are better process control and automation. So a lot of the processes are still very manual, which means that standardization and variability can become an issue. Hiring staff with cell and gene therapy experience is challenging. These are things that aren't taught at university that you really need to be doing it in order to learn it. So it requires hands-on learning. There's a requirement, so for more commercial CMO manufacturing options, as well as more clinical options. So people are having to schedule very far out in order to get somebody to help them make their cell therapies. We need to have better analytical testing for both cells, and even though we don't use them internally, vectors and viruses, to ensure that what's going into these products meets all of the guidelines. And modular manufacturing facilities would be very useful because it would mean that companies would be able to reconfigure very rapidly in order to meet different needs. So if you need a whole bunch of different types of molecules at a smaller scale versus a single molecule at a very large scale, being able to reconfigure for that rapidly would be very helpful. So overcoming some of the safety and commercialization barriers is going to be very important. So, this is particular to immuno-oncology, but being able to replace viral vectors in order to increase safety profiles and reduce cost. There really aren't any good factories for making autologous cell therapies. So these are therapies where you're having to do the same process over and over, but you have to do it each time for each patient. So, having processes in place in order to do that effectively with kind of that stringency that we discussed earlier would be very helpful. Bringing point of care manufacturing closer to point of care is really important. Transporting cell therapies from where they're made and getting them to patients really can be quite challenging. There's a lot of controls that have to be in place, so being able to make them closer would be very helpful. Not having allogeneic therapies is a huge challenge. So that ties back to number 2, where we're having to do the same thing over and over for each individual patient. Having some off-the-shelf, in quotes, options would be very helpful. Another thing that's challenging is that the research really is kind of outpacing what can be done in terms of scaling up and bringing things into the clinic. And what we really need are new technologies that are going to do what Synthego does, which is to bring high throughput scalable platforms and really apply them to the cell and gene therapy world. So how are we doing that? So we're really working to improve ribonucleoprotein delivery so that we can get away from having to use viral delivery. Reducing off target with novel chemistries and also using bioinformatics to help predict what kinds of off targets might be a problem. Really trying to develop this end-to-end cell and gene therapy manufacturing platform. So I kind of alluded to that when we were talking about Halo and Eclipse platforms and how they're going to combine to make the next platform, which we envision as being these cell and gene therapy. So being able to make cell therapies for people, and in doing so, decentralizing the manufacturing. So having something modular that can be brought closer to the point of care. And then I think that we're really getting close to the end here. So currently, what we're seeing is that GMP CDMO capabilities support clinical trials and diagnostics. Is we have GMP grade guides. Where we kind of want to be as we're moving forward is we want to be able to provide these therapies. So we want to expand our ability to make GMP guides, and we want to get that next platform up and running so that we can do GMP CRISPR deliveries and make therapies so that to help alleviate some of the kind of bottlenecks that people are facing now. And that, with that, what I kind of want to say is that, by enabling these platforms, by bringing these cell and gene therapies closer to the patient, what we're really going to do is address the fact that patients are waiting, and many of them really can't afford to wait very long. So solving these problems is very important, and it's something that we take very seriously. And with that, I'd like to ask if there are any questions that we can address, and thank you for your time.
Awesome. Well, thank you very much, Abby, for the presentation. Thank you to everyone for joining. And yeah, just drawing attention back to the last slide here. If you want to get in touch with Synthego, please feel free to reach out to support@synthego.com for any technical questions, or if you are interested in these GMP topics specifically, get in touch with Abby, who can either help you directly herself or put you in touch with someone else on our GMP team. Speaking of which, I do want to draw some attention to a comment in the chat from Ethan Brooks, who is our director of clinical sales at Synthego. In particular, he recommended that for anyone in the US to have an INTERACT meeting early on in the development process. INTERACT standing for Initial Targeted Engagement for Regulatory Advice on CBER Products. I know a lot of you in the audience had some questions regarding the regulatory landscape. I'll let you read Ethan's comments in more detail, but I do want to point out the last comment he said, which is that it is critical not to wait on this until you get to the late stage, or your trial could be delayed possibly by multiple years. So definitely good to get a head start on regulatory conversations, and in terms of sourcing material, definitely good to get in touch with Synthego early on in the process. So with that, if there are any other questions, feel free to post them in the Q&A box or in the chat. I'll give it a minute for any of those questions to roll in, but Abby, any final thoughts that you want to share?
Yeah, I agree with Ethan that like getting your interactive engagements going early is really important. We always say, speak to us early. Even if you're not ready, even if you're not sure what it is, what you're going to need or exactly when you're going to need it, starting these conversations as early as possible is going to be very important in ensuring that you have a supply chain in place, not just for guides, but for everything else that you need in order to bring your therapy or diagnostic into clinical trials.
So I see someone posted in the Q&A box here. What do you mean with PD cell batch?
That would be a process development cell batch. So that would be cells that have higher quality that you can use towards, and are going to be very similar to, in terms of quality, would be very similar to what you're using in GMP. So it's meant to kind of make that transition from research in the clinic. It allows you to do your development. It would have some documentation with it. It's kind of a cell line equivalent of what we're calling GMP-like guides.
So kind of an intermediate step, so to speak.
Mm-hmm. Yep.
Awesome. So I'm not seeing any other questions roll in here. If there are any last ones, just 1 or 2 minutes left to post them in the Q&A box or in the chat. Here we go. I'm not sure how much of this you can discuss live on the call right now, but Abby, someone is asking, how much more expensive would it be for GMP sgRNAs? Are there any ballpark numbers you can give, or does that require more individual consultation?
Yeah, I would say that really depends. So some of the things that really are going to impact price are how much guide RNA do you actually need? So how much do we have to deliver to you? How do you want it vialed? So some of the testing that we do or that is required are based on a percentage of the number of vials that are made in the batch, as well as what kinds of testing do you require for release, as well as any stability testing that you might do. So when we take all of those factors together, that enables us to calculate how much guide we have to make, as well as which will enable us to price that out. So it's really hard to take a guess at that without more knowledge.
And Abby, I'm sure you could provide more details on this, but from what I understand, it's really important for customers to understand when they are talking with vendors, the difference between whether or not they're getting quoted on the amount being synthesized versus the amount being delivered, right?
Yeah, so what we base on how much do you need and what are your tests rather. So I think the way other companies are doing this right now is you say, I want you to make a scale of this amount. So you want a gram scale. And then you tell them what testing you want, and then you end up with whatever's left. So what we're trying to do is tailor a little bit more towards what do you actually need, because that can help bring some of those costs down. It also means you don't have a whole bunch of guide RNA that you've paid for that you don't actually need and won't ever use.
Yeah, for sure. Cool. Well, I did see a few last minute questions roll in here, but we're already at the top of the hour. So if you do want to chat in more detail, if we didn't get to your question, or if you're curious about how Synthego can help with your particular research project, again, feel free to reach out, either support@synthego.com or your local account manager, like Abby, for example, in the GMP space, or myself, if you're doing more discovery side things in the Boston area. But in any case, definitely check out Synthego's website. We're very glad that you could tune in for today, especially if you tuned in for the other sessions that we had earlier in the week. I believe our marketing team will be sending out the slides and recordings to all 3 sessions within a few days. So no worries if you Didn't get to tune into all three. But with that, once again, I want to thank everyone for attending. Want to thank Abby for speaking, and definitely want to say thank you to my colleagues for helping put this together. So take care. Bye.
Thank you, everybody. Have a good day. Bye-bye.